we would like to get into our first session of the CDRC annual meeting. This one will be on rare diseases. You can see a list of the moderators and panelists we have here today to discuss all of the pertinent topics that we brainstormed before. So the moderators are Matt Might and Shira Strongen. And we also have Andy Krause on the panel, as well as Ethan Prosing, Eva, pardon me, Eva Morapa Kovic, Lisa Schill, Omid Karkoti, Claire Thibode, and Sandra Simone. And so if you see a star also by the panelist's name, that indicates that there's a presentation for the session that they have pre-recorded. Those presentations are also available on the website, and you can go back and watch those if you haven't had the opportunity. They're extremely informative and pertinent. And so with that, I'll pass it over to Marco.
Thanks, Chandler. Thanks for going through those housekeeping items, and I'll have to say I was thoroughly impressed with the speakers that provided those presentations. So thank you very much, speakers. If you haven't had a chance to view those, I highly recommend that you do. And so I'm not going to spend too much time. My name is Marco Schito. I'm the executive director of the Cure Drug Repurposing Collaboratory. This is based out of the Critical Path Institute. We're a nonprofit organization. This is an effort that's funded by the FDA. We work very closely with FDA, so I think that these types of conversations are really instrumental in being able to identify, address, and have those communications surrounding the issues, not only with rare diseases, but with other diseases as well. And obviously, that's what we're going to be talking about today. And I've asked both Matt Might at the University of Alabama and Shira Strongen, who's actually at the FDA as well, to lead some of these discussions. And we've been having some preparations over the past month or so to really dig into some of the challenges and be able to have this conversation in a way where we're including the scientists, we're including clinicians, patients, and advocates, as well as individuals who have interest in this. I know that there have been lots of interest in the communities. And I appreciate those who have registered and come online from different parts of the world. Actually, I looked at the registration list, and there's people from all over that have signed up. So, with that, why don't I pass it over to Matt and Shira and let them introduce themselves as well as the panelists.
All right. Thanks, Marco. I appreciate the opportunity to co-lead this with Shira. For background, I'm the director of the Precision Medicine Institute at the University of Alabama at Birmingham. I'm also a rare disease dad, and I'm extremely passionate about personalized therapeutics, trying to find the best possible treatment for any given patient, and especially rare disease patients. And I've found over the years, working both within the context of my son's disease and the broader rare disease community, that repurposing has a major role to play in this. And that's why I'm so excited about this panel and the panelists we have here today. So with that, it would be great if my co-moderator, Shira, could introduce yourself.
Thanks so much, Matt. My name is Shira Strongen. I am currently a senior at George Washington University, and I'm an ORISE fellow at the FDA's Office of Medical Policy, working specifically in drug repurposing. I've been there for about 3 years now. It's kind of a wild ride. I'm a patient myself with a rare disease whose life was saved by drug repurposing. So it's critical for me to be a part of these conversations and to be able to add that voice and that perspective. And I'm so grateful to be able to work with people who do value that and who value the patient perspective in this. And I'm really excited for this panel. We've put together some really wonderful speakers for you. I hope that you've had the chance to look back at the YouTube videos and recorded speeches ahead of time, but if not, no worries. Most of the presenters will be summarizing their talks within these, and the questions will be able to add some more information there as well. But without further ado, I think we can go ahead and get started, Matt.
Absolutely. So let's go ahead and begin with some panelist introductions. If you could introduce you, well, there was a slide, but I'll call it out in the order it was on there. so that we can get to know a little bit more about you. And please consider this a chance to advertise your talk as well. So I know some folks have watched in advance, some folks haven't. So if you did post a talk online, please tell us a little bit about what's in there so we have some context for some of your discussions today. So Andy, if you can introduce yourself.
Sure. My name is Andy Kraus, and my background is in biochemistry and molecular genetics to start, and I've kind of taken a winding path out of academia through industry, biotech area for about a decade, and then I moved back into a nonprofit that was a genome institute in genomics, where they started focusing on the diagnosis side of rare genetic disorders. And as they began to get really good at that, from my perspective, who was not familiar with this area at all, at first I was, interested in what, the families were so gracious, is so just really found benefit from having the diagnosis. And then my question was, what next after that? And so at first I did, because I really didn't appreciate just how much the diagnosis meant. And as I came to appreciate that, and we really moved in that direction, I then saw Matt Might come and speak at Hudson Alpha, where I was, and he kind of came with the path forward for what next after that. So at that point, I reached out to him, and now I'm the director of research at the institute where he directs at UAB, which is the Precision Medicine Institute.
All right. Thank you, Andy. Ethan, if you could introduce yourself.
I think he's late.
Yeah, I believe Ethan was having some issues with technology. So if we want to go ahead, and Eva, you can introduce yourself and maybe give a little introduction for Ethan as well.
So Ethan and I worked together on drug repurposing just about before COVID. We linked together and started to work together in these really difficult times. I'm a clinical biochemical geneticist, and he's a PhD. He did his PhD at Harvard. I'm Hungarian, actually, and I was only recruited by Mayo 3 years ago. Ethan has been working on screening for new therapeutic candidates in worm models, and came up with a few target drugs in different disorders. And I have been working with patients with different rare disorders, but my major focus is CDG, congenital disorders of glycosylation and deglycosylation. The disease group has been discovered 40 years ago, and this year is the first year that we have the first clinical repurposing trial for the most common type of CDG, PMM2 CDG. So these are historical times, really. Ethan found new drug targets, and we got very excited because this is a new chance to have a rapid success in our patients. Unfortunately, the road to clinical trials was not easy. We were successful with a single patient IND. We saw very promising improvement in our patient, and we have been working on trying to get a clinical trial submitted to FDA, but the disease is rare. The patients' numbers are low. The patients cannot wait to get on therapy after 40 years. It is really hard to convince them to do a placebo-controlled trial. And obviously, there's difficult to make power in a small group. So we are facing many challenges in our pioneer work. But just to give you a little more enthusiasm, I just saw this new drug we were talking about in our video was tried in a fish model. And the fish model showed that the fish will drop on the bottom of the tank after a couple of days because they have this genetic disease. And if you add the drug to the water of the tank, they revive and start to swim again. So, you know, when you see in an animal model such an effect, then you just cannot wait to start and try to treat your patients. All right, I hope I wasn't too long.
Oh, that's perfect, Eva. I really appreciate the context, and also a reminder that we are entering sort of unprecedented times when it comes to repurposing and rare disease, and really every disease in general. There's been a lot of progress in a very short amount of time recently. With that, Lisa, if you could introduce yourself.
Yes, thank you, Matt. My name is Lisa Schill, and I'm a patient advocacy and engagement consultant. I am also a parent advocate. I was formerly the vice president of the Resopathies network. The Resopathies are a group of genetic conditions caused by mutations of genes in the RAS-MAP pathway that affect cell growth and cell regulation. The Resopathies include Costello syndrome, Noonan syndrome, cardiofaciocutaneous syndrome, and neurofibromatosis type 1. 20 to 30% of cancers also lie on the RAS-MAPK pathway, and we believe that repurposing existing RAS cancer treatments could be beneficial to the Resopathies. Recently, selumetinib was repurposed for the treatment of plexiform neurofibromas in NF1 and FDA approved. Currently, MEK inhibitors and mTOR inhibitors are being prescribed through compassionate use for hypertrophic cardiomyopathy and lymphatic issues in Noonan syndrome and Costello syndrome. It has been an honor to be involved in the CDRC Rare Disease Working Group to improve repurposing for all rare diseases. I look forward to contributing to today's discussion, and thanks so much for having me here today, Shira, Matt, and Marco, and team.
Well, thank you, Lisa. Omid, if you could introduce yourself.
Thanks. Hello, everyone. My name is Omid Karkouti. I'm the co-founder and chief operating officer of RareBase, a public benefit biotechnology company based in the San Francisco Bay Area. Our focus is rare and ultra-rare disorders that fall beneath the threshold of typical biopharmaceutical investment. And in particular, the proceedings in this conference is of particular interest to us because we work together in collaboration with patient communities representing the rare disorders and patient advocates to identify, discover both repurposed as well as novel therapies that may be brought into their given indications regardless of its prevalence. I'm really excited to be part of this conversation. Thank you.
Thank you, Omid. Claire, if you could introduce yourself.
Thanks, Matt. Hello, everyone. My name is Claire Thibode. I'm the Vice President of Scientific Affairs at Cures Within Reach. Cures Within Reach is a nonprofit based in Chicago that is focused specifically on supporting repurposing clinical trials. So we've been doing this for over a dozen years now. And where we like to focus is the pivotal catalytic clinical trial that will then be able to produce enough data for PIs to leverage that either into additional follow-on funding or publications that will allow clinicians and patients to look at the data and decide if this is a treatment they want to try for off-label use. Right now, our current portfolio is 33 projects in 21 different diseases across 8 countries. We are disease agnostic, but about over 40% of our current portfolio is focused in rare diseases. And we work with our supporters in a donor-directed manner to focus on the areas that are most interest to them specifically. This year, we have 3 new initiatives that we're very excited about, one focusing on pediatrics, one on veterans, and one in the diversity, equity, inclusion space, which includes not only repurposing research to address health disparities, but also supporting underrepresented and underserved researchers and clinicians doing this work. So I just want to thank the CDRC for putting on this event, and we are very excited to be participating today and at another panel later in this in the event. Thank you.
Oh, thank you, Claire. And last but certainly not least, Sandra, if you could introduce yourself. Oh, I think you're on mute. Nope, can't hear.
Sorry. Okay.
No.
I am sorry,
Sandra
Sandra.
Petrosian Sermoni, and I am the CEO and founder of the ADNP Kids Research Foundation. Can you hear me?
Yes, we can hear you.
Okay, sorry. Now you guys are all echoing. I am first and foremost mom to an amazing little boy who has ADNP syndrome. ADNP syndrome is a fairly newly discovered syndrome, and it is a neurological genetic syndrome characterized by intellectual delays, neurodevelopmental delays, autism, sleeping problems, feeding problems, GI problems, brain and heart abnormalities, and it pretty much globally affects the kids. The interesting part of it is that it is a top single gene cause of autism. And also recently this year in a paper was discovered to, they believe it makes up approximately 0.2% of all autism cases. It's also linked to Alzheimer's, Parkinson's, and schizophrenia. My talk today was to explain our experience in working with a repurposed drug for my son's rare disease that we actually found through our artificial intelligence with Matt Might, and why we believe that this drug, which is called ketamine, is going to be therapeutic for ADNP and others with idiopathic autism. Ketamine is got a little bad rap because it's known as a party drug. So we're in the middle of trying to get past some of the hurdles, but we have cleared our phase 2A FDA drug trial recently with stellar results, and are now considering what we need to do moving forward to get this drug faster to our children than the typical FDA phase 2, phase 3 route. I'm here to discuss different opportunities that we may have by basketing autism syndromes together, perhaps sharing costs. Right now, the burden falls on these small organizations with probably 50 families contributing to fund everything. And I just want to have more discussions and find better ways to move these repurposed drugs. Ours is generic, so we have a really great benefit because it's so inexpensive that we think we might have some ways around wasting millions and millions of dollars doing a phase 3 drug trial.
All right. Thank you, Sandra. It's definitely exciting progress. And once again, I want to encourage folks, if you haven't seen these talks, they're really outstanding. I would absolutely encourage you to go online and check them out. Not right now, after the panel, of course. And given the panel, I actually have a long list of questions I want to ask, but yet another one actually popped up while I was listening to everybody give their introductions. So I'd like to throw this out there. Ava, I think you really described it well when you said that these are kind of historic times. And I know that you've been involved for much of your career in the search for therapeutics for disorders of glycosylation. And I feel like there's sort of a sudden acceleration in terms of repurposing and real progress. So the first question I want to ask, and this is really to anybody here, is this acceleration real? Is it really happening? And if it is, what's driving it? What's actually causing this acceleration in the drive for repurposing?
Parents.
That's a great answer. Well,
and it is accelerating. I mean, Matt, if you know this, but others don't. From the time that we heard about ketamine, it took us about 4 to 5 months to pull enough clinical data to file IP and to create a hypothesis and to present it to Mount Sinai, and another 5 or 6 months to get FDA approval. So in less than a year from discovering this, we started an FDA trial. In less than a year from that, we completed that phase 2A FDA trial. And so now we are blowing straight into the phase 2B, perhaps even before we publish. So we're doing this at a much faster speed, you know, where we can possibly get our children a drug in, 2.5 to 3 years after discovering it.
So can I ask you how you've been so successful? Because you will be my muse.
No idea. We have really good luck. We did this all in the middle of the pandemic too. Like literally started this in the eye of the pandemic.
Is it a placebo-controlled trial or a safety trial?
The first was a safety trial, 10 children, open label. The really good results though that we had from that was, the parent goggles are always on when they know their children have the drug. But they all saw significant improvement across all domains. The good factor was that so did the clinicians who were using clinician rate tools and scales. They also matched what the parents were seeing, and that was with one single dose.
And how long was your trial in the first, the safety, the open label?
The kids went in for 5 weeks.
Wow. And what was your outcome measure?
We had many outcome measures. The most kind of important are, we had all the neurodevelopmental speech, repetitive behavior, daily living, all of those autism and neurodevelopmental sort of outcome measures, and they showed improvement across the board. But what's best is that it showed our preliminary data has showed a 2-fold increase of ADNP expression. So our children have one functioning allele. It is bringing that allele up 2-fold, which is basically making up for the one that's not functioning.
But what was your primary outcome measure? I'm sorry, I'm like interrogating you, but we also submitted an observational trial in 9 patients with CDG and just observing clinical outcome measures, and we failed through the FDA. They asked us to do a placebo-controlled study in 9 patients. There are 9 patients in the United States with this condition. So we were forced into changing our trial design and randomized the 9 patients to 2 groups, which will just kill our power. I know it already, but they just stated that there is no possibility to do a trial without double-blind placebo control in a rare disease.
That is our next phase, and we're going to use approximately 30 children, and we'll have 2 groups of 15 each. And it will be a placebo-controlled crossover study. So in the second half, all the children will get it. That's the plan. Again, it's not submitted yet. We're still working on it. And again, I'm not the doctor or the PI running it. I'm just a mom who runs the foundation and who helped find ketamine. So the better person to speak about that, I can connect you with.
Mm-hmm. So I'm really impressed, and I'm really curious, how is it possible that you and I submitted almost the same trial design, you guys with the rare disease, but a little more patients potentially, and we on a patient group, which is maximum 9 patients in the US, and you got away with the observational design, and I got clinical hold for longer than a year, stating that without placebo control, they are not supporting my trial.
Well, we went in it straight. We went in it first as a safety and efficacy study. So I think that may have helped us. And then we also threw in our outcome measures just to see like what kind of data can we collect. We were super impressed. We didn't expect to see such incredible data with a single dose. But the main structure of this phase 2A was safety and tolerability, and efficacy. So, will the kids spontaneously combust when we give them 1 dose of ketamine? We know they wouldn't, but we had to prove to the FDA that they wouldn't. And then we also did our outcome measures, and we learned a lot, which I can't discuss because it's being written on a publication, but the preliminary data looks spectacular.
Sandra, I know you say you're not a PI, but gosh, you're starting to sound like a PI. And I find this a lot, and you credited, parents, and I think in general, of course, we patient advocates and patients in general really are driving a lot of this in a way that they haven't before. It's very clear, listening to you that is the case. And Ava was really starting to dig into some issues that affect a lot of us when it comes to running clinical trials. I mean, a lot of these communities are ultra rare. In some cases, 9 patients in the U.S. could be a lot, and so I think it starts to get to a more general issue of how do we deal with the fact that some of these patient communities are so small? How do we deal with the issue of needing to run placebo-controlled trials when we know a priori we won't have the statistical power to get to statistical significance? It just isn't going to happen with communities this small. Is there a need to do more with natural history? Does the FDA need to get more adventurous, more creative in terms of data science? So there's a lot of questions, but I think are in scope for any number of our panelists. So if anybody wants to jump in on any of those, questions of regulatory flexibility or small patient communities or doing better with natural history, feel free to jump in.
Well, and we'll kill our entire clinical population with our phase 2B. So that's what concerns us about our phase 3, because we'll kill the whole ages 5 to 12. We'll use them all up on our phase 2B. So that is problematic for us. And this is why we're trying to think outside the box of, what do we really need to get the drug faster to children? Our sort of idea right now is that, an FDA phase 3 drug trial is there to get approval so the insurance will reimburse. Our drug costs $20 a month. So do we really need insurance to reimburse a drug that costs $20 a month when the foundation could probably give stipends out for that or scholarships? We don't have millions of dollars for a phase 3 trial. Can we pull together $500,000 to do a very detailed placebo-controlled crossover multi-site study? I've got Black Friday on my mind. To do a very detailed treatment study, yes. And our thought is that most doctors can write off-label drugs as long as it's been de-risked. So we're trying to de-risk it. We're trying to describe it. Our drug this is an ultra low dose. I think there's probably less narcotics in a year's supply of what we need to give our kids than the bottle of Vicodin they sent my daughter home with her knee surgery. So it's very low risk. We just happen to have one that's a narcotic, which goes against us, but it's also generic, which it goes for us. So we're wondering, what information can we maybe get to what do you need to get those physicians writing that drug for those patients and following those patients, even if their insurance doesn't describe it. And then also we believe we should be doing basket trial studies. We've got, SYNGAP, FOXG1, FOX P2, Phelan-McDermott syndrome, all these other rare diseases that are very similar that we know that some of them, some of these, we have a list of probably 25 genes that have a reduction in the ADNP protein. So if this drug increases ADNP expression in good alleles and they have functioning ADNP genes and just a reduction of the protein, this could be therapeutic. So why can't we lump us all together in a basket, use all those resources and outcome measures and these specialists who are here to design these trials and just basket out each one of us separately, but all share the expense. So there's a whole lot of things I think that we could do to save money. But, for those from the FDA on this call, parents are paying for this ourselves. And we can't work jobs because we are taking care of profoundly disabled kids. we can't afford million-dollar drugs, multi-million-dollar phase 3 drug trials. So there's got to be something else.
So
Well,
I think we're
similar
gonna,
to what we're
If
doing
I
at
could
Rare
just,
Base.
yeah, I think Sandra brought up a really important point, and I can talk about this from Cures Within Reach as a funder perspective. You know, when she said, what's the data that we need to convince physicians and clinicians to think about these repurposed drugs in an off-label setting, especially when we're talking about generics. And at Cures Within Reach, you know, we are open to a lot of different study designs because we know, especially in the rare disease community, it's not a one-size-fits-all approach. And we have had success with that in the past in rare disease, where we've had small patient populations and we've looked to natural history or as a comparator, and our PIs have published data. This was a project out of the Children's Hospital of Philadelphia for ALPS, a rare condition, and this particular PI that we supported was able to publish his results in a journal. Other clinicians started contacting him, asking about how he treated his patients, and this was using sirolimus, which of course is a generic drug, and eventually became adopted as a standard of care. And it wasn't because there was an application to the FDA, it was because there was a pivotal study followed by case reports, followed by other clinicians confirming the results and inquiring about it. So I think it's really important, especially when talking about rare diseases, to think about different ways that we can look to clinical adoption, because as Sandra has so beautifully told us, you know, it's about the patients. Well, you know, it's about the kids, it's about the adults who have been suffering from rare diseases, and how can we create that patient impact. So thinking about different ways that we can design trials, that we can use real world evidence, natural histories to support that process. And the FDA, as part of the 21st Century Cures Act, has looked into that. They have started to have a policy on real world data and real world evidence. And I think more connections with the FDA, whether it's through the CDRC or other opportunities for patient groups and researchers to learn about how they can use this data in an appropriate way for the FDA, would be extremely helpful for everyone.
Absolutely.
I'm glad to hear that.
So much for bringing that up. As the resident FDA person on here, while I'm a moderator, so I will not be primarily participating in the panel, but I do think that this is important to address, is the project, the Cure ID project that we are involved in with CDRC is all about collecting real world data to inform clinical practice, clinical trials, and label changes as well. And I just want to affirm that that is a priority. It is something that we're looking at, and it's something that we recognize needs to happen, particularly in the rare disease community, where there isn't necessarily the same funding, same patient population numbers to go through traditional processes. And that's what the CDRC is looking at. We're looking at different ways to support patient populations to make sure that we have the access to the therapeutics we need. So I really appreciate all of you being here today and having these really important discussions. I am going to turn it over to the chat because we're getting a couple of questions in there. One in particular is what is going on with cancer repurposing at this time. And actually there will be a later session specifically on oncology and repurposing. So I invite you to attend that session as well. And there's a specific question again for Eva, which is, was the dose for your 9 patients also an ultra low dose of an existing generic drug?
So you will probably get surprised that we got on clinical hold for this trial, because the actual drug we use in this specific CDG is galactose, which is a monosaccharide, the major component of milk sugar. In this disorder, the patients who have glycosylation disorder use this supplement since we reported improvement of a single patient in a case report. So exactly happened what you guys described. There was literature proof that this simple sugar added to the diet has major effect including endocrine, hormonal, coagulation, blood sugar levels, like a multi-system improvement in this metabolic disease. So that was followed by other case reports, and that was followed by a small pilot. And then all the patients were placed on this treatment. So when we tried to do a proper clinical trial, and we said, all right, so we would like to do a safety trial on 9 patients and a dose escalation. FDA requested us to do a washout and do a placebo-controlled a blinded study. And so, first of all, it was disappointing because this is galactose, so you have it every day. You get, as a newborn, 3 grams per kilo in mother milk. The second reason it was disappointing because doing a washout in these patients who are already on the drug could cause life-threatening problems, hypoglycemia, bleeding. And that brought me into a big ethical, moral crisis. And I didn't have the chance to discuss this with FDA because they didn't accept my request to talk to them. And so I'm not sure where I'm not doing this right because my study design was very similar to what you described, Sandra. This is like not a sedative, it's not a dangerous drug, but it is dangerous to take it away from patients who are already on it for several years. So I'm not sure, Shira, if you can give me any good feedback and suggestion, because in metabolic world, most of the patients self-treat, and some of the treatments are working quite well, just never ever been on a proper clinical trial.
And I will be completely transparent, and my work is with Office of Medical Policy, so I don't work in anything to do with drug approvals. I specifically am looking at drug repurposing and different pathways to patient access there and different ways that we can determine efficacy as well. So that is not my area, but I'd be happy to help connect you with those who might have better answers within the agency. So we can chat offline about that for sure. I think it's a very helpful dialogue to have at this kind of event, because I know it's not something that is necessarily discussed as much in the patient population community. We talk a lot about what drugs we're taking, but we don't talk about what it can take to get them approved and that very lengthy process. So perfect, Andy, you actually answered the question that I was just going to point to next. Thank you so much. Andy, if you want to address it out loud, there was a question in the chat that was talking about the process of identifying ketamine using AI. And I know Andy and Matt were the people that were helping to do this, so I can turn this over to them.
Sure, thank you. Happy to do that. Yeah, so this is a process that has been developed over the last 4 years plus. It is a very large project that's funded by NCATS. It's called Translator. And the effort sounds monumental, and because it is to effectively unite all biomedical knowledge into one place where you can start to really increase the value of any scientific data that's out there already. And by connecting all these dots, you increase the value of every data point that's out available now. So you can have things where you might go to PubMed and you would search for one gene relationship, and you would get one paper, you'd have an A-B relationship, and then another paper might have a B-C relationship going on. And if those are A, B, and C are genes, and you said, tell me about what the A and C, how they're related, you would get no answer because those are disconnected data points. And so by connecting those two publications together, you can actually really get to new and novel data just by looking at what already exists. You can start to develop hypotheses by looking at what already exists. And so in this particular case, we have a process for working with families like Sandra, who I'm just so impressed. She's like, I'm not a PI, and I think we're going to have to really define what just a mom means for sure. 100%. Because I feel like, there's a quote in there. I don't know that she knows what that word means. If it's just a mom, and then she proceeds to talk about all the science and everything, it's amazing what you've done. So families like that, we can work with them, and we really go in with a hypothesis that says, okay, we think that the gene was impacted this way, therefore we need to upregulate the gene, or we need to downregulate the gene. And we can use these artificial intelligence tools to get at those relationships that are out there. We can ask basic questions like, what drug regulates that gene and how is it regulated? And so in this particular case, we found that our tool was returning ketamine as an example that's out there. And that was what we come back with. Now, what we've really developed a process around is, how do we work with families? How do we do this in a way that we're on the science side of this? How do we get that back to the people that either can do more science, because that is very often the case for these answers. It doesn't always, jump straight to here is a drug, but we can find places where there's either new research that should be done. We basically bring it right up to the edge and say, we need a researcher to answer this question in order to do this. Or if we have something like in this particular case, we can go back to the family's physician and say, okay, here are some thoughts, here's some ideas. Let's educate you on this issue and see if this is a thing that you might move forward with. And so in those particular cases, after we talk to the physician, they get to go back and decide to practice medicine with their patients. So we have a whole yes, well.
Well, that's kind of the nature of repurposing. You never quite know what's going to come up. And so I mean, you could head down any number of clinical and regulatory pathways depending on the answer you find and how you get to that answer. I mean, it really just kind of gonna have to wait and see what happens when you get there. But yeah, Andy's right that by connecting all this data together, it's interesting the new suggestions that are coming out of it. And I think this is one of the accelerators that's having an impact in terms of repurposing. And actually wanted to touch base on this and some other topics that came up earlier about the challenge of running these trials. And it's challenging if you're a patient community and you don't have enough patients. But there's also the emergence of these new commercial players that have these this incredible mission to go out and help these patient communities. And I was curious, Omid, if you've had thoughts about these challenges as well. How do you do repurposing for ultra small communities or even ends of one? Is there an approach that industry can help us with here?
Thanks, Matt. As we've got some students of statistics on the line, I'd love to hear that be a part of the discourse. But I think that when we start thinking about statistics empowering a traditional clinical study, or when we start to get into pharmaceutical repurposing, we face the challenges of incentives for industry to participate in the area because the drugs belong to likely someone else, or the incremental increase in patients who can be prescribed a medicine from the originator of that medicine is likely not going to change the bottom line. When we think about patients that could potentially benefit from a drug, I'd love to put the question back up to the group in thinking about if you have a very, very highly variant response to a drug, could we better measure the effects of that in an individual, or measure the progression of the disease very sensitively in the single patient and individual? A really easy way to think about it is if there's a patient and then during the course of their disease they go from 1 to 5, and then a second patient in the course of the disease they go from 10 to 50. We would never be able to really, we're looking at that highly variant population. We would not we couldn't take an average of those two patients and come up with something that is within an amount of variance that's acceptable. But if we could very closely monitor the progress from 1 to 5, we could see that if the intervention kept the patient from progressing from 1 to 5 and instead stayed at 1 or went from 1 to 2, we wouldn't need to compare it to 50. And so thinking about the nature of the looking inward and looking at the individuals and their progress, can that be, and I think that's part of the big part of the discussion here, can that, and leaving the efficacy of the efficacy part when we think about pharmaceutical repurposing to the real world setting, so long as the intervention is deemed safe, is probably the only way for these disorders where there aren't sufficient patients to power a controlled trial, a placebo-controlled trial, a randomized trial. I mean, you just never can get to the numbers based on standard statistics. So that's what I think a big part of this question is that how do we engage the regulator in enabling that type of thing? And so love to hear your thoughts on that. There's lots of technology that is coming up, engagements of patient communities, additional diagnoses that are finding people that don't take no for an answer and are looking for solutions wherever they can. So how can they best start to measure or enable the measurement or enable clinicians to start following their patient communities using technology, using validated scales, using what are the best practices to do so? That's going to be where we get the data that allows us to apply pharmaceutical repurposing, and will be important to enable pharmaceutical companies to also contribute in this.
Yeah, I think you've raised some really critical points here that in some sense, with numbers so low, we do have to amplify the data within individual patients. We do need to collect more real world data, real world evidence, in addition to the usual natural history. And I think what's exciting is that there are, through technology, better ways to do that than ever before. So I think that this could become a major player going forward. And I wanted to pivot a little bit along the lines of some earlier thinking in this direction too. Sandra brought up this concept of basket trials, one drug, many different diseases. And is one way to get around, in some cases, the issue of small numbers. I feel like if there's ever a community that could pull something like that off, surely it's the Resopathies. So Lisa, I don't know if not to put you on the spot, I don't know if you can comment on the state of basket trials for Resopathies, or where the community might be headed in that direction, and any thoughts you may have in that regard.
Yeah, I would say that the Resopathies Network has been thinking about this for a really long time by bringing all these, smaller syndromes together, that looking at a pathway approach. We'd love to see a basket trial. I think a lot of our researchers aren't as knowledgeable on basket trials, so they need more information. I also think that the FDA, since 2016, with using real world data, could give more guidance on what can be done. So we look forward to seeing more guidance as the years go on. But I don't even think it's just bringing all the diseases together, but also looking at all the different endpoints. We've seen that there could be a possibility with lymphedema, with hypertrophic cardiomyopathy, and also, improving cognitive deficits. So, we don't want to just pick one biomarker or the easiest one, measuring the thickness of the heart, So, how can we best approach this? I'm so thankful that we are bringing all the stakeholders together. I mean, I really wish that we had payers here as well, involved in these conversations, because I think it's, really important on, how we are going to get these issues solved, and even bringing together the clinicians that are using MEK inhibitors and mTOR inhibitors off label through compassionate use. They're all kind of running around to find out, what is the best dosage and when should we give it and how long should we give it. And the FDA has been collecting this data, what can we do to help incentivize the FDA and also clinicians to share this information with one another. So I'm glad that I'm here so we can all start to discuss this to really make a difference.
So Matt,
in the real world data too,
Yeah.
that Matt was talking about, there's also things like biomarkers where, if your child's missing a protein and you've just increased that protein, is that real world data that could be used? Because, as we all know, neurodevelopmental disorders and profoundly disabled kids are very hard to clear the FDA. Like 95% of those clear the FDA. So can we start using some of that real world data on visual potential studies, or or RNA protein increases. Can any of those be used when you have a safe drug to start earlier than later? Because you talked about these discussions having for years. We don't have years to have these discussions. We have a drug now, so how do we get that drug in our kids as fast as possible?
Can I just ask something? Sorry.
I was just going to make a point about the real world data. And Matt, you mentioned the technologies that we can be used to collect this real world data, whether it's online patient registries or an app like the Cure ID app, which allows physicians to post their case studies, or wearable technology, those sorts of things. And those are all really important. And I think one of the great things about real world data is it gives patient populations who might be underserved or underrepresented in the medical community the ability to participate in research in a way that they haven't been before, whether they're based on geographic or socioeconomic issues. And I think that when we're talking about those kind of technologies too, it's important to remember that not everybody has access to those technologies as well. So I'm thinking about ways to make the data set, when we're talking about real world data, representative and equitable as well, will strengthen any clinical trials that are based on that real world data, or any clinical trials that involve that real world data as well. So again, that's just another factor to keep in mind. I know that when we're talking about rare disease, it's not the same as when you're talking about more common diseases in the population, but it's still important that representation does matter, and the results will be stronger for it.
Absolutely. Thank you so much for bringing that up. I do, since you brought up Cure ID, just want to share a little bit more about that. It was initially designed for infectious diseases. However, it is being expanded into rare diseases with this group, which is very exciting. And the majority of the people that are on this panel are actually part of the efforts to drive that. As we discussed earlier, we're actually going to be piloting it within the Resopathies and are looking for other communities as well. So if anybody who is on this call is particularly interested, please do reach out to myself and we can get a dialogue going and see how we can best support you within that, because we do recognize that getting all the clinicians together and having those conversations and making it very easy and accessible is important, but also having patient opportunity for feedback and parent opportunity for feedback. So currently we only have a clinician reported outcome form, but within the rare diseases, we're actually looking into developing a patient reported or parent reported outcome form as well. And to turn it back over to Lisa, not to put you completely on the spot again, but I think it would be interesting just to hear your perspective, since the Resopathies are, the disease group that we've chosen as an initial pilot study, and hear your perspectives on this.
Yeah, I think it's really important. Like I was just saying earlier, I see, a bunch of the researchers sending messages to each other when they have a really sick baby in the hospital and they know that they're not going to make it, and they want to try, to use one of these repurposed drugs as a last ditch effort, and trying to find this information isn't readily available. And also, should we be waiting to use this as a last ditch resort, or should we be using, the mTOR inhibitors and MEK inhibitors earlier on? So I think once we get all this data together, it's really going to help make clearer decisions for the clinicians, but then also help the patients and their families make better educated decisions about what are the risks and benefits of using this.
Yeah, and Lisa, you bring up an important point. You brought it up earlier too. I think every panelist has hinted at this to a degree, that there's already a tremendous amount of experimentation happening by default within the patient communities. And that's what's really, to me, exciting about efforts like Cure and Cure ID, to be able to actually start capturing all of this sort of natural experimentation that's going on, and hopefully structure it and expose it in such a way that we can then extract signal from it and say, hey, it looks like this might be working. This is something that's worthy of more serious investigation. For really for everybody on the call, what I'm curious about is, where do you see this heading? Is it practical to start, collecting? What would it take to start really collecting all this information in a more structured way, such that we could do that? Our patient community is willing to engage on this. How far would they go in the terms of, collecting that data? And then for folks more on the side of, using that data, how might you use it? How might we start to see the signal that we could use in arguing for the efficacy of a drug for a given condition?
Yeah, Matt, one thing I will also mention on this is that, to capture this real world data either through an app or through other means, it's hypothesis generating, which can drive clinical trials, obviously. But the interesting part, and Claire brought this up earlier, is the 21st century cure, because here the FDA are thinking about utilizing that real world data to be able to supplement a clinical trial data. So Sandra, as an example, if you've done a 2A study and we also capture some long-term as well as some other data that supports the results that the clinical trial has using real world data, knowing the fact that it's going to take you years and years to be able to do a clinical trial, and the difficulty around utilizing the limited number of patients to be able to get statistically significant results. Can we leverage the data that's already out there to be able to supplement that information so that from a regulatory standpoint, the regulators can make a judgment call based on the level of evidence that's being generated. So that's one thing. And then the other question, I think, Sandra, that you pose as well, is that do we need a regulatory approval? So the whole point of getting a drug FDA approved is the process, right? You've gone through this process. You've said that the drug is safe, it's effective, and you've got data to show that. But that's being used by a company in order to sell the drug. So it allows them to sell that drug for that indication. In this case, do we need, I think we need to go through a process to make sure that, yes, this is going to be safe and it's going to be effective. But if it's a repurposed drug that is generic, the sponsor really doesn't come in. It's not really up to the sponsor, because the sponsor is saying, well, anybody can make this drug. It's off patent. So is there another potential way in which we could address this to actually benefit the patients and benefit the prescribers? And that's really the question that I'm struggling with, is to try to find a way to do that. So from a clinician's perspective, how do we engage to make sure that clinicians know what to do and where to go and what to use? And I think that's where this potentially could lead a road down to be able to identify that sort of pathway, where potentially it could be guideline development, but I know that not all clinicians look at guidelines. It could be, when we talked about labels, they said, well, that's where a clinician could potentially go. If it's on a label, then they'll use it. But I've asked clinicians and they say, we don't look at labels. So, how is it that clinicians actually decide on which way to move in these different aspects, whether it's a rare disease, whether it's a cancer, whether it's all these other things? And that's, I think, the challenge here is that there isn't really a good source, and oftentimes it's based on where they work. So if you're at the Mayo Clinic, you have one source of data. You're going to go to the Mayo Clinic, and there's a place where if you have some questions, you can ask, and it's really a circle of colleagues. And if you're in the Cleveland Clinic, it will be something completely different. And so I think it's a challenge on doing that. That's why guidelines were developed. But I think just because with everything else, it's very difficult to consolidate a lot of these things in one area. I think that there are some resources that we could explore a little bit further, some online resources, as well as some groups that are trying to consolidate some of these things. But I was just wondering, I think, Eva, from your clinician's perspective, what are your thoughts on that?
Thank you. And I actually wanted to comment a little bit earlier already that you guys had successful trials and you had small patient populations. So probably you know the trick. But if there was somewhere a toolbox collecting these successful approaches, and rare disease clinicians or scientists could approach or could have access to that toolbox with examples of successful trial or study designs. For example, CureID could have such a tool garden. I think that we could use that approach and learn from the successes and would be much quicker in designing the right study or trial setup. And the same what you just said, if we could share off-label use successes, even if it's publication, because you cannot follow all the publications in all different, from autism to immune deficiency. If there was some kind of library where you could read about these, that would speed up so much. Like what I just heard and learned about resopathies and autism today, that is relevant for my patients too. And for example, there are some autism issues in my patient population. So maybe, that can be applicable for my patients group as well.
So
Yeah,
if I
and
can,
actually one of those other things, just before you get, sorry, Claire. David Fagenbaum, who's at University of Pennsylvania, and I don't know if you know him, Eva, but he basically discovered his own cure. He had a rare disease, Castleman, and discovered his own cure using sirolimus, and that's what's saving his life. And I think Shira has her own story about the repurposed drug that she's being used to save her life. But these are the type of things that that are starting to become a little bit more well known. And I know that the Castleman network, the thing that David put together, actually has a roadmap initiative that is doing just that. So we're partnering with them to be able to identify all the different groups that are out there that are pulling this off, and to be able to say, what are the lessons learned? What are the clinical trial designs that worked for you? So to be able to pull that, as you said, that toolbox together. So I think that this is being done, and we're partnering with them to be able to do that. Sorry, go ahead.
Also,
I
It's
was
not
going to say,
pediatric friendly though.
so also one quick thing just to interject very quickly, and heading over to Claire after those, actually what you mentioned, Eva, is being done at CureID. So we do have a database of literature reviews of what is happening, of case studies that are all there in addition to the clinician reported outcomes that can be submitted. And as we develop Cure Rare, Cure Pregnancy, all of these other things, that format will continue. So that actually will be available when Cure Rare is live for rare diseases. And there was another message in the chat similar as well, of a registry of all drugs currently being tested. And that is what we hope to have with Cure Rare, similar how we do with Cure ID, where you have a list of a variety of different diseases, all of the different drugs that are being tested, outcomes, whether they are successful or whether they were unsuccessful, and whether patients were harmed or not. All of that will be available there. So that is currently being developed, and it's really exciting to hear that this is what our community wants, and that's what's being worked on. And so I know Claire has been very patient, so I would love to hand it over to her.
Yeah, thank you. I was just going to say one other aspect to add to the toolbox, and this is something that, I don't think gets discussed enough, is funding. Because we can design a really great clinical trial, we can have top candidates, and if there's not funding to do the work, it's not going to go anywhere. And Cures Within Reach, we are able to fund a certain number of these kind of trials, but there needs to be more awareness of funding that's available. For instance, through the FDA for orphan diseases, they have funding grants for that. There needs to be more funding focused on clinical trials through NCATs or other sources, looking at repurposing, not just in the AI space or in a preclinical model, but in clinical trials. And I think that the more that we can do to expand the funding opportunities, Matt opened with this question about, is this a real trend that we're seeing in repurposing? Is this really something? And I can tell you, we've been doing this for over a dozen years, and the amount of applications we get from PIs in all across the board in various disease areas has been increasing steadily. More and more PIs are interested in repurposing as an avenue. More and more patients are becoming aware of repurposing, especially with COVID-19 and what we've seen going on. And so now, it's never a question of, are there enough repurposing ideas out there? It's, is there enough funding to get them going and get them off the ground? So I think more efforts to increase the pot of funding for repurposing that's out there, and more resources to direct PIs who think, I have this repurposing idea, I don't even begin to know where to start for funding. It would be a really valuable addition to the toolbox and to future developments in advancing repurposing research for rare disease and beyond.
Well, yeah, we could use that immensely, because we don't know where to get funding. Everything that I've described, we as parents have funded 100%. And like the real world data that you guys are talking about, we're trying to thoughtfully think of how to best spend our money. And that would be to run a complete FDA phase 2B, because then that literature's out there for everyone. It's not just a case study someone has to hunt down from the Mayo Clinic. It would be an FDA, it would be, I'm assuming, a positive FDA phase 2B, as our phase 2A is probably going to be published. So, but we're funding that 100% ourselves, which to me is crazy, because A, we're one of the top single gene causes of autism. B, we believe that this is going to, and the scientists have all said it publicly, this is going to treat a large array of idiopathic autism. So this is like the first autism drug, and it's $20 a month, but it's generic, so no one's helping us fund it. So it's falling on our shoulders. So that funding is a huge thing for us.
Yeah, and I'm Claire and Sandra, I'm glad you both brought up funding. And at the risk of turning this into a group therapy session, I did want to ask the other panelists, to what extent is funding a problem, especially on the PI side? Where do you find it, and is there enough out there? Ava, I don't know if you've had issues getting funding for the trials that you're working on or not.
For the patients, they are amazing, and we get the most funding from them. FDA FDA, NIH did fund our time to set up the trials, but they are, in at least the grant mechanism we are using for our patients, doesn't fund the patient cost, only the trip is covered. So that's very difficult, because you blackmail your patients into the trials, which is ethically totally absurd, right? You can come and I take care of you, but all the other costs will be yours. Like, that's also already a big bias. Like, who can come and get the therapy? Probably a lot of people are unable to get access to therapy. So I totally agree with Claire, and I'm sorry that we didn't bring this up earlier, but this is a huge problem. And I'm not sure if you guys are smart about this and know about smaller grants for this kind of smaller trials.
Ava, my son, I helped push ketamine through and did all the research and pitched it, and my son can't even be in it because we can't afford to come from Washington State to New York for 5 weeks. So I found the drug. My foundation paid for the whole drug trial, but my kid can't be in it because I can't afford to travel. So yeah, there's so many different aspects of funding that you have to think about. It's not just funding the trial. You have to get families there, or you can only have families who can afford it.
Yeah, and part of this gets to, the way we do clinical trials right now, having everybody go to one site, as opposed to maybe leveraging technology to make it more distributed, and make clinical trials more accessible and frankly more equitable too. I think there's real equity issues when it comes to clinical trials. And I think that's kind of what we're trying to get at with real world data and real world evidence, is to lower the barrier to actually making these critical discoveries. We talked about the issue of patient communities being too small, and how perhaps capturing this additional data might allow us to still be confident in our decisions. I also wonder if the current system is just too binary. It's on the label or it's off label. It's either or. Maybe we need degrees of flexibility in between. Maybe there's a drug that is officially promising but not yet proven, which can serve as a signal to clinicians and maybe also to payers, that this is something they should pay attention to. I mean, clinicians, it does seem in many cases, are willing to be more flexible when it comes to the use of drugs off label. Payers, much less so. I think payers are much more hesitant until there's more data. So one question I want to throw out there to folks is, as we design these trials, the FDA is an important consideration, of course. We'd love to see these drugs get on label. I'm wondering though, can the real benefit here actually come in the form of convincing payers that these drugs are worth covering? And is there something, do we want to think about this as we move in the direction of using real world data and real world evidence to think about drugs?
Yeah, I know somebody, a mom named Amber Freed, who has a drug right now that she spent a lot of money on doing repurposed drug trials and has found something. But that drug, I think Horizon owns it. It's $800,000 a year or something astronomical. No funders. They can't probably fund it for their own study. So, we're very lucky. And this is why I really push more of this technology to try to find these gems like ketamine. But, there are people like Amber who found something and now they can't access it because of these astronomical expenses. Like, who's going to help with that?
And
I think there has been, oh, sorry. Go ahead, Eva.
just a very short remark, the placebo price is also very high. So if you want to do a proper blinded placebo-controlled trial, making the placebo safe and be sure that it doesn't have an effect on the disease phenotype can cost a lot too. Sorry for cutting you off.
Oh, no problem. I was just going to say that, I think because of the issues with the cost of drugs and the cost of having a rare disease, that they're the reasons that the Every Life Foundation and also the government has started some studies on the real cost. What does it cost to have a rare disease? And I think that, these studies are timely because it really shows the cost. So even if there is an expensive treatment, it might be that it's less expensive than using something else. And as for real world data, I also like that we're starting to use the genotype first approach. Some hospitals like BioMe is using it at Mount Sinai and Geisinger Health to really start sequencing people as they come in the door and looking at their electronic health records to see when they are diagnosed with this rare disease, what their symptoms are. And I think that that's going to change the course of how we see diseases as well.
A small comment, just circling back to what you said, Matt, is that, and something that Marco said before, if the FDA is primarily in the business of keeping people from marketing poisonous snake oil, let's say, if we reduce that, keep people from keep anyone from trying to sell something that doesn't work and or primarily isn't really bad for the patient communities, and the payers are perhaps the barrier with which we need to develop a data set. And I would hate to see the FDA label be an excuse not to pay for medicine that could have a significant benefit to patients, and then it isn't accessible because of that, because you need to pay the retail price of the example that Sandra shared, a drug that the manufacturer does not wish to have the label be expanded, because that would cause negative price pressure on that product. They don't want anyone else to take that drug because they have a locked-in patient population, and that's an economic thing. You can find it in their financial reports as a public company. They don't want it to be cheaper. They want it to be more and more expensive. So what is the barrier? What is the MVP data package that a payer can use? And some of the studies that you were referring to, really, Lisa, the studies and publications that have come recently and talking about the cost of living with a rare disease can be used as an economic basis, perhaps, to help a payer understand. But ultimately, if the FDA label is an excuse, and the product is an excuse not to pay for or reimburse a treatment, then we're in a really, really hard position if the regulator is difficult in enabling trials to take place, even with something like a dietary sugar. So yeah, I mean, I know I'm just rattling off different problems, but I want to focus in on that. What can be the solution? What's that MVP data package that a payer can look at? And then folks, a lot of dedicated and advocates and folks on the line that I think are super smart and could think about what that could be, and happy to participate in any of those following conversations. Is like, can the payers be addressed? Because for repurposing, if drugs can be prescribed off label, then the data can get out there in a manner like a clinical study that a patient organization funds, like ADNP-Kids. If that drug was unaffordable, that would be a really, really difficult thing. That would be putting data out there and a payer doesn't agree. So I'd love to hear what the group thinks about that too.
I would just like to add, I mean, even though people are getting approvals through the accelerated approval process, payers are still now sometimes not paying because it was through the accelerated process for rare diseases. So that's another issue that, another roadblock that we're facing as well.
Yeah, so I think, whether it's on label or not, payers still have to make their own decision about, what they'll cover. But there's, I realize actually there are certain drugs, like for example in epilepsies, which I don't think have ever been properly shown to work for epilepsy, and yet are so widely used they are covered by payers. And so it seems to me that some payers are using an implicit real world data standard in that regard when it comes to deciding what they cover. I'm wondering if we should try to figure out, at what point does a payer think, okay, I'll just cover an off-label drug because it's so widely used that, and it's sort of just known to be working for a particular area that they really do have to cover it. So there's, to a certain extent, it is happening, and maybe there are ways to pick up on that signal that's out there. Yeah, and
I think that's a role that the CDRC can play, is helping to bring payers to the table, because we can sit around and discuss what we think a payer may or may not need, but what would be really beneficial is to start that dialogue with payers so that we can better understand what they're looking for, what they need, and then in addition to all the other factors we've described, kind of keep that in mind when it comes to as was mentioned, the MVP data package that's needed. So the more that we can do to bring payers to the table and have these conversations with them and start that dialogue will be beneficial for all the repurposing projects, not just rare, but beyond that too.
And I'll just mention as well is that this dovetails nicely with the regulatory session that we'll have tomorrow afternoon, by the way. And David Simon, who's in the audience here, virtual audience, and I have been thinking about this as well as a number of others, and I've had some discussions not only with FDA, but with groups in Europe, in the UK, in Australia, and the list goes on, of other groups that are starting to, we're not the only groups that are having these issues. It's global, right? And all these other stringent regulatory authorities and some of these others that may not be so stringent are exploring ways in which that this could be done. So for example, I'll just point out to just put a plug in. A lot of it is obviously cancer and the oncology field does sort of push the envelope sometimes. Oftentimes they do. To address formularies in which, and that's how many of these insurance companies figure out whether something is going to be covered or not. But the other aspect of this is, and you can hear, I think it's Sandeep Agrawal. He has a presentation if you want to go on session 4. And he talks about Project Renewal, which is a way in which FDA by themselves goes out, takes a look at the drugs that have been off patent for the past 20 years and have all of these clinical trials and pulls all that information together, does their own review, and then informs the group that holds that drug to let them know that FDA is doing this for repurposing, and they bring them in on board with it. But it's all work that is being done at FDA. For the purposes of identifying new indications for old drugs. And these are types of pilot projects that are going on in a variety of different groups, both in Europe, in the UK, and having a lot of these discussions in Australia. And I think that we can learn a lot as if we can come together as a community for rare disease, but come together as a community for regulators to be able to better understand how best that we can support the communities and understand, how we can work with the various stakeholders, including payers and industry. So I'll leave that as a plug and let you guys discuss further.
Well, I think it's become very clear that there's a huge role for payers here. We have to get them involved when it comes to repurposing, or all the efforts of the FDA will be for naught if payers aren't on board with this. I did want to take some time to talk about repurposing in a broader context and to see if we've arrived at a moment, sort of indirectly because of COVID, where there's a broader understanding of repurposing in the public consciousness, and this might be something that we can use, but also to be mindful of some of the pitfalls that have come with some of the very open calls for repurposing within this. Of course, there's been successes like dexamethasone, and that went through sort of the process that one might have expected, randomized controlled trials and seeing benefits. Then there's been less regulated efforts. There's been calls for repurposing of things like ivermectin and hydroxychloroquine and bleach. I think this has muddied the waters a little bit in some sense. And so what I'd like to know is, are there lessons to learn here? And I will say that it was interesting for us at the Precision Medicine Institute early on, because we were doing some of the early drug screening in collaboration with our partners like Kevin Harrod, testing some of these compounds. And we actually saw, at least at a cellular level, some evidence of efficacy for things like hydroxychloroquine and ivermectin, if you're willing to take them at very unsafe levels. And that was sort of the caveat we saw, which was, well, yeah, they sort of work in a cell, but they're probably not going to work in a human. In fact, when it comes to ivermectin, there's a lot of diseases it can treat if you're willing to take it at unsafe levels, or so it's been our experience. But that's something that has to get communicated, I think, clearly to the public, is that there's a difference between a cell and a human being. And so I just want to ask the panelists, what can we learn from the examples of COVID, both the successful ones and the ones that have generated so much controversy?
Well, Matt, I think you hit the nail on the head when you talked about, communication of clinical trials. So it's, on the one hand, it's really great that the general public has now a better understanding of repurposing and what it is and how it can be done. As you mentioned, there's dexamethasone, there's baricitinib, there's most recently fluvoxamine, which are all repurposed successes to treat COVID. So those are great examples for people from the rare community and others to point to and say, this is what I deal with every day, trying to be in this crisis mode, finding therapies for myself and my family who need them yesterday. So, you know, that's the pros of where we are with repurposing right now. The cons is that the general public still doesn't really have a really great understanding of research and clinical research. That, you know, we can see stories that come out in the news that say, you know, ivermectin is great in a cell, and they don't understand that then you still have to go ahead and do the clinical trials. So I think the lesson is keeping the message of having appropriately designed clinical trials that you use as facts and data to then make treatment decisions. And I say appropriately designed because when you're talking about COVID, you do the phase 3, thousands of people, confirmatory trials, which isn't going to happen in a rare disease. So again, getting a study that's well designed for the patient population and for the outcome measures that can be looked at by clinicians, by payers, by FDA, and say, okay, this is something that you know, we can all accept this treatment as a standard of care for this population. So really understanding that when you're in the world every day, these might be things that come second place, or second nature. But when it comes to communicating to the general public, you know, communicating those sorts of ideas is still complex and requires finesse on all of our parts. And it's a great message to share. We just have to make sure we're sharing it in the best possible way.
Very much agree with
And
that.
some of those drugs, like you said, were, you know, they're high doses. They're not safe doses. What about those drugs like ours that we know are a safe dose? You know, how do we speed that up and not have to go through some of the bells and whistles that, you know, when we've already proven it? It's already had 30 years of data that shows that it's safe in children. How do we speed that process up? Because for us, our children, time is brain.
Yes, absolutely. Yeah, go ahead, Andy. Yeah.
I was just gonna say, I'm wondering in some cases like that, you know, that particular drug in general, if there were a way to mine the EHR data that's out there. So we've done projects where we've actually over the summer, we published a paper that showed that we found that if you're a diabetic and you are taking metformin, then your risk of death goes down. It's almost like you're a non-diabetic in that case, and that signal was just kind of hiding in this all of the data that's out there in the EHR. Nobody would have just put that together potentially. It really did take taking a huge swath of data that's out there, distilling it, asking a question about it, and testing that hypothesis. So something like ketamine, if there were a point where we could go in and find enough patients that were on this or children that were on this, and you could start to show how safe it was even outside of the population of rare disease, that might be another way that you could get at some of the safety of this. That's not my specialty to know like what is the safety and if the comparison of someone with a disease and without is a real valid comparison. So I apologize if that's overstepping there. But I also wanted to go back to one of the things that sort of I think that is another really important piece in this in terms of what lessons that should be learned from COVID is absolutely communication. This idea that people come out and say, and part of the problem will be the press, because they're going to want the headline and they're going to leave off in cells, right? You're going to say, this stopped COVID in cells. And if you've ever talked to somebody, a reporter, they get the byline, right? It's theirs. They'll have your quote and then they will interpret it. And then that's what's going to go out. And so it is very hard. So I think the opportunity to speak in front of people, if you know, we have people from UAB that did a great job being on CNN during all of this. And we really just have to stay away from, you know, really just couch the statements very carefully, because if you say it works or it doesn't work, or the differences between it doesn't work and we don't have the evidence to show that it works. Those are two very different, like in one case, it sounds definitive as if you've proven it doesn't work. And in the other case, we're still looking at this, we're to decide if it works. And it's just a very different concept that the nuance is lost on, you know, very often when it gets filtered through the news. But I think those two things in terms of one, how to speed it up, and then how to communicate it back to the public at large. And I think I could imagine that, you know, these different rare communities are, you know, they're all a microcosm of what happened with COVID, you know, to some relationship. We have to, when we speak to them, we probably need to speak to those same lessons and have that same nuanced communication as well.
I was just going to add, I just don't think the narrative is done yet, right? You know, Merck and Pfizer are going after emergency approval for some drugs that were dormant therapies that were sitting on the shelves. And I think if we, you know, once COVID, hopefully one day is a thing of the past, we can look back and really see the narrative of, you know, how much money had to be invested by the government for something to be done. We can look at, wow, these were potential targets. Some of them worked, but most of them didn't. And I think it's the same in rare disease. And so I think it will be a good way, you know, once we're almost through this, to look back and say, see, this is what the rare disease community has to go through. But the difference is we have smaller population size and we have no funding. So this is why it's important. And I think more people will understand that as we move on.
I also think from the communication side, absolutely, I completely agree. But the other aspect of this is that there was pretty much a lack of coordination, primarily around COVID, with getting this. I was just struck and amazed by that. Just everybody did this. It was just a starburst that everybody just went off and did their own thing, and as a result, you had hundreds of COVID trials, you know, being used, or hydroxychloroquine being tested for that. All really, really small, all completely underpowered to answer the question. And so there was a lot of just, you know, waste of money, waste of time, waste of effort, and primarily due to lack of coordination. We saw that differently in other parts of the world. In the UK, where you had sort of the one-payer health system, that they could really be able to hone in on that. So I think you know, our public health has been, the system has been declining just simply because I think that they've been doing actually a really great job over the years. The problem is that they've been doing a great job. So, hey, they don't need as much money. So it's been neglected for probably another 20 or 30 years. But there's also this other aspect of it that I wanted to bring up, is that there was this huge rush of having publication. Every journal was looking for, you know, what data is out there, and send us the data as quickly as you can, and we will publish it as soon as we get it. There was very little review process that was put in place. A lot of those were just put online and made available because everybody just wanted to have some information, and not going through it and critically reviewing it and identifying areas of issues that that should have been caught that weren't. And as a result, those papers were pulled up. The press got it. CNN ran these different articles, and then you had an explosion of people saying, "Look, there's a repurposed drug, and we're not using it because nobody's making money off of it." So, you know, conspiracy theories then just blew into that. So I think from my perspective, that's how I saw that you know, some of the lessons learned is through those types of analysis. And I think, you know, going through this in hopefully when we get to the other side of COVID, we can take some of those lessons and ensure that they don't happen again.
Yeah, Marco, you bring up a good point that, you know, if ever the stars had aligned to make repurposing go smoothly, surely it should have happened with COVID. I mean, you have an essentially unlimited patient population, sadly. I mean, you really do. And many proposed treatments, many with good rationale, and many which still have not been tried. And yet we had, you know, dozens of trials on hydroxychloroquine, you know, all too small, underpowered, and whatnot. We're still sitting on, you know, suggestions that we're still trying to get into clinical trials. I think we launched one clinical trial total of 20 some repurposing suggestions that we were able to come up with. And so even with COVID, there's been a lack of coordination, strangely a lack of funding even to launch these clinical trials. And it's been deeply frustrating because you think, you know, if ever there was a time to get this right, this was it. But it also highlights a challenge for rare disease communities. You know, so AI can make a lot of hypotheses really fast in some cases. Yeah, I think when we did our initial repurposing screen, it generated 40 suggestions. 20 of those ended up actually working as antivirals in cells. And there's now evidence that some of them are even working in people, either through our own efforts or through the efforts of others. But if you flip that around to a rare disease population where you have a very small number of patients, it's not inconceivable that it may generate more drugs than patients as hypotheses. And so I want to put this out there. This doesn't always happen, but when it does, how does a rare disease community go about the process of trying to prioritize what to try when it comes to the suggestions that are coming out of not just AI? There's lots of approaches that can generate interesting suggestions for what you might do that then need to go through some kind of proper vetting, but possibly in a very small patient population. So, Sandra, I don't think you face this. Lisa, I don't know if there's a larger number of suggestions out there for resopathies or not. And the same for Ava, if you have more suggestions than you know what to do with, or if there's issues with ranking that you may face.
Well, we were lucky. We had clinical data. There's publications already on animal models, so we were fairly lucky on that.
I would be interesting to take a couple of these cases and because I think what I've heard is the pros and cons in both directions. You've got the cheap generic that has a huge impact, but then the downside is it's a generic, therefore, and I almost wonder if it wouldn't be very worthwhile to take those scenarios that the different groups have run into and write up a paper on it and just, outline, here's the pros and cons. I mean, just because I would be shocked if anybody could go into this and just foresee, all of these things that would happen. And I think it might be a really interesting paper to capture that and get it out in the world so that the next person, because again, I think going back to Matt's initial question is what are is it really accelerating? To me, it would feel like the more people doing it, the more people will do it, kind of thing, that there's going to be just barriers that are knocked down and knowledge that's gained through all of this. So I would propose that would be a fantastic paper to put out there to sort of get through these nuanced descriptions of a couple of different case studies in a way of the roadblocks that are there.
I think for our researchers, 20 years ago, the RAS pathway was considered undruggable. So there weren't a lot of options sort of out there, and the safety profiles weren't very good when you're talking about, repurposing a cancer treatment for the regular population. But, as time's gone on, the safety profile has improved. Things have gotten better. There have been issues with, I know one specific drug a researcher really thought would be the best out of all of them, but didn't have access because the sponsor wouldn't give access to that drug. We had another drug that was tried in mice and had, a really good outcome that he was convinced we were going to, move it into people so quickly, and did a trial, just a study, not a clinical trial. But the results weren't there. And it, was it dosage? Was it the time that it was given? Because we're talking about germline mutations that, happens in every cell of the body from the time you're born. Do you need to give it prenatally? So it's really complicated in the resopathies. It's not as easy, I think, as some of the other syndromes. And then, for the most part, the resopathies is tapping down, putting on the brakes a little bit of the pathway. So, it's going to be interesting as we move forward on which avenues, we take on what to use. And the only way that they're using them right now is in life or death situations. So how can we move from the life and death situation to, using it more, taking that chance to use it a little bit more regularly, because we're talking about doses that are 200 times less than what you would give in a cancer patient.
Another thing,
I think the life and sorry.
Come on, Sandra.
I was going to say, I think the life and death flexibility is an issue as well, because children like my son have an incredibly difficult life that gets worse as they get older. And I don't think that's accounted for when you talk about life or death. Perfect example, my son got a Make-A-Wish, and they heard his story and looked at him and saw how he was, and they actually slipped and said, it would almost be easier if he had cancer, because you either cure him or he doesn't live like this for the rest of his life. So I don't think people understand the severity of what life is like and the quality of life for these kids. And just because they're not going to die doesn't mean they're not equally as important. I think that's
Oh, it's hard to say anything after this, Sandra. Oh my God, I love you. You're a wonderful person and a wonderful mom, and such a fiery wonderful person. Anyway, I wanted to bring in outcome measures because I think that all these great ideas could fail if we potentially think of basket trials or different Resopathies. You know, we try to use the same drug, maybe the drug of action would affect all these different disorders, but our outcome measure might be different for each disorder. I don't know if anybody worked on basket trials before and has like a genius idea how you pick your primary outcome measure when you put together different diseases, even if they belong together, but they present differently. Anybody has a good suggestion?
Thanks, Ava. The one thing I will mention on a couple of things. So, you know, I think Andy mentioned earlier about the potential of using electronic health records. So we've got FDA was able to secure a grant to actually automate the extraction of data elements from electronic health records. And we're working now with variety of different groups, including Johns Hopkins, Emory University, University of California, and the virus registry out of Mayo, actually, and Society of Critical Care Medicine, to be able to not only develop the tool, but to embed it in those institutions and multiple different institutions, not only within the US, from around the world. And we're working closely with the ISRIC database out of Oxford and the Infectious Disease Data Observatory, who run the database for COVID-19 at the WHO. So that's definitely a larger aspect of being able to ensure that we not only automate that extraction, but that we build in a leave-in infrastructure so that once that's there, we can then make, rather than just look for COVID as being the diagnosis, we could look for any one of these diseases. It could be a rare disease, it could be another infectious disease, it could be a disease in pediatrics, that then utilizes it as an infrastructure. And then what we're hoping is that if these are presented and implemented in a community setting, that allows then clinicians and physicians in those community hospitals to be able to enroll patients and then use the electronic health records to be able to supplement the data that's being collected. So that's one way in which we can think about doing this. And we've spoken with a group that was just recently stood up a couple of weeks ago called ACT at POC, which is the Coalition for Advancing Clinical Trials at the Point of Care. And there's definitely an interest to be able to utilize these sort of leave-in infrastructure at community settings to diversify the patient population, to ensure that you have more inclusion in the patient population, and to be able to allow more of platform and basket trial designs to be implemented. So I think those are things that we're really excited to be able to work with a variety of different groups that are coming together on this, and you know, to ensure that we have patient populations, that we have the advocates, that we have the moms, the dads, the clinicians, the physicians that are all being a part of this, because you know, we have to come together as a community in order to do that, but we need the infrastructure to do it. So we're hoping that this will be able to something that we can do. And I'll stop there.
All right, thanks, Marco. And I noticed that we're getting close to the end of our time, so I wanted to check in with Shira to see if we'd missed any questions from the audience and that we still have time to answer.
We actually have unintentionally answered most of the questions in the audience, even though we didn't bring them up. They just happened via dialogue. But if anyone does have a final question that they want, please feel free to put it in the chat or just specifically in the Q&A, and we can share it. I think something I would be interested in bringing up is, Sandra, you've brought up so many incredible things today, but one that really stood out to me was the beginning when you talked about how you were lucky, right? That you're lucky that all of these things happened. And a theme that I've been hearing over this panel discussion is that we need to make sure that this isn't reliant on luck. We need to create pathways. We need to make sure there's access to data. We need to ensure that we are all effectively communicating as stakeholders, from payers to patients to clinicians to researchers to regulatory agencies. And I would just love to hear from each of you if you have an idea of what that ideally would look like for you.
So before you answer that, Sandra, the one thing that I took away from this meeting, I think, Andy, you actually said it first, and that is thinking about writing a paper on all these case studies, because as I go through and I talk to different groups, I get a lot of the naysayers, right? Oh, drug repurposing, that doesn't work. When does that ever really work? And all of this. So, to be able to, and that's what I've been thinking about doing more and more, is to be able to, even just create some slides to be able to have that as in the back of my mind, but actually put a manuscript together to show the various different success stories. There's not a lot of them, but they're becoming, as Claire said, more and more people are getting into it. You're starting to get more and more success stories, but each one of those have different lessons learned of why they quote unquote became lucky. Like what happened in order for them to really move the needle from a really basic concept to all of a sudden I'm in the middle of a phase 2 clinical trial, right, Sandra?
Yeah, well, we did. I mean, when I say lucky, it was lucky from the Matt Mike perspective. I don't know if anyone watched my presentation, but I slightly stalked him for many years. And we were lucky to get something. Matt Davis and I, though, had to put in an incredible amount of work. I literally went to multiple drug development conferences, learned how to pitch, even getting coached how to pitch this. There's so many different facets, though, that this is what I would like to let rare disease organizations know, is that these, like I said, I'm not a doctor, but these are things that you can do. The most difficult for us is the funding. I mean, we can fund the beginning of these small trials, and our families rally, and we have amazing supportive families. But getting the FDA involved, getting the NIH involved, even getting on a phone call with the FDA has been physically impossible for me, and I've tried. But once we
I'd love to help you get in contact.
Once we've proved ourselves, right? How do we get help on that? I'm almost a one-man show here, and funding I am not good at. But how do we get the FDA or Simon's Foundation or CZI or one of these big organizations? When I say lucky, I mean lucky like this is not just for ADNP. I can almost confidently say that we believe wholeheartedly that this is going to work for numerous types of idiopathic autism. So, when you have this huge lucky drug, when I say lucky and generic, how do we bring in the funders to help us move it forward for the greater good, because they're not going to make any money on it?
So, Sandra, I think that, in the same way that it was amazing to me when I was at the diagnostic with a group that was doing diagnosis, and Matt came along and was like, he blazed the trail in some ways and captured it as a process. And that's what we do today with our cases that come to PMI. And then I feel like have taken now what's next, and if that's why I think the paper or something that would codify that to help the lessons learned, like you said, pass on to other people would be really a fantastic idea. And I mean, it's the blood, sweat, and tears that you put into this are amazing. And, the lessons learned would just be, I think, invaluable to be able to capture that and put it.
And
funny thing that Matt told me when I first called him and told him, we got FDA approval to start our phase 2A. He was like, what? And he said something to the lines, I mean, you correct me, Matt, but like, he's like, this is exactly what this program was meant to be. We'll find something and then hand it over to the patient organization. And then it's up to you guys to like do all the research and get it all done. He goes, but I just didn't believe it would happen this fast. And I just kind of teased him and said, that's how we roll. And like I said, we have been, we have a second drug in the pipeline. And now we also believe the ketamine combined with another drug is going to be a better drug than ketamine alone. So, to have three different sort of opportunities in less than 6 years time, 7 years time from being discovered, and spending very little money. I think the maximum we've spent so far to date, and we've done a phase 2A drug trial, is about $550,000. Think about that. We have all those drugs in the pipeline. So I think we found a way to utilize the parent power. I always say, like, a parent is the most motivated machine in medicine, a rare disease parent. So I think a lot of it can be learned, and that's why I really want ketamine to be an example for that, because I know some of these are going to be a lot harder than something like ketamine, but if something like ketamine comes on board, the parents and the patient organizations can do a heck of a lot more than they think they can.
And I think this is a great example of the continuum of research. And I think, thinking back about, how can we make this happen more smoothly, is building that continuum of research, and, how it goes from AI to cells to humans, to the proof of concept, safety, to the phase 2, to all of that. So, we have a translational approach when it comes to new drug development, but we haven't yet applied that in the repurposing setting in a structural kind of way. When I give presentations, I always say there's Department of Oncology, Department of Neurology. There's no Department of Repurposing at universities or other organizations. And a lot of times you'll have a preclinical study that shows promise, but then it stops there. Or maybe a mouse study, and then it doesn't go anywhere. But the more that we can do to make those connections and build that community, that continuum, and build that pipeline to push forward, we can create a structure that where repurposing can happen much more quickly and efficiently in the future.
Claire, I think you sort of, that was a beautiful recap and forward-looking question to end exactly on time. That was fantastic. So since we're at time, I just want to thank all the panelists and my co-moderator Shira for contributing today. This was a lively discussion. It went by fast. There's clearly a lot left to cover, and I'm excited to see what comes from this, in particular the shape that toolbox begins to take. So I want to thank you all and see you all at the next session.
Thanks, Matt. Thanks, Matt and Shira, for moderating this. And yes, it's only the first series of volleys, and we'll hopefully revisit this in the future as well. Thanks, everyone.